this poster, we presented the prediction peptide-protein complex structures with the Alphafold2. Actually, predicting the structure of these kinds of complexes is going to be one of the most important and amazing research fields, specifically for designing peptide drugs and enzyme inhibitors. A lot of computational algorithms and tools have been designed and developed till now, but most of them need to have crystallized protein or peptide structures. And since obtaining these kinds of structures is too expensive, so using these tools also has its own limitations. Nowadays, some sequence-based complex prediction tools such as Alphafold and RoseTTAFold are becoming more popular. And actually, in this study, we analyzed Alphafold for predicting peptide-protein complexes with both template-free and template-based methods. And we tried to evaluate and score its result to figure out how accurate they are. For this aim, at the first stage, we designed a pipeline that starts with obtaining peptide-protein complex samples from PDB websites, and all these samples were released in 2021 and 2022 years. But since most of these complexes have similar structures, we apply city-hit algorithm to generate non-redundant datasets with a sequence identity cutoff of 0.4, which means we removed sequences that have more than 40% similarities, and we consider the final 12 complexes as a dataset for our study. In this poster, we just presented the prediction process for two samples to show template-free and template-based results here. We selected 6L02 sample to show the template-free method, and actually after predicting the peptide-protein complex structure, we align it to the native one to find out how accurate it is. For predicting the binding site in 6L02 complex, we defined interacting residues if their minimum distance is within 5 angstrom. The red color in this table indicates the binding site on both protein and peptide sequences. And then we tested template-based methods of Alphafold on another sample which calls 7 RNS to predict its binding size. We did it the same way that we implemented template-free method. We aligned the predicted and native structures together and also calculated the distance between residues of protein and peptide to find out interacting parts. To evaluate this process, we compared the predicted binding site with the native ones and defined a formula to calculate the accuracy of the prediction process. For calculating this score, we divided the number of intersection binding sites on both predicted and native complex by the union of them, and calculate the percentage of accuracy for this score. And actually, we calculated this score for both protein and peptide sites. Based on the results we have obtained in this study, in some cases, the template-based methods outperform the template-free and vice versa, and in some cases, both methods perform the same. Among all 12 samples that we tested, Alphafold just couldn't predict one of them very well, that we indicated with doesn't predict well, it's 7DZ9. Overall, we think that it could be a good idea to use both template-free and template-based methods together to have a more accurate prediction process, as they are complementary methods. And also, since Alphafold can predict a complex structure very close to the native one, we can use this tool for a wide range of research fields such as designing peptides or peptide-protein interaction studies and analysis. This study was supported by the National Institute of General Medical Sciences, of the National Institutes of Health, and also the Bond Life Science Center at the University of Missouri, Columbia. Thank you.